Good morning. My name is Nizar Jarjour. I am the division head for allergy, pulmonary, and critical care. And on behalf of the Department of Medicine and Dr. Lynn Schnapp, who couldn't be with us today, it gives me great pleasure to welcome you to the Department of Medicine Grand Rounds. This morning, it's my privilege and honor to introduce our guest speaker, Dr. Wright. Dr. Wright is the Goldsmith Professor of Pediatrics and Environmental Medicine and the Dean for Translational Biomedical Research, as well as the Director of the Institute of Translational Science at Mount Sinai Health System. She's also the Director of the Institute for Exposomic Research at Icahn School of Medicine at Mount Sinai in New York City. By way of background, Dr. Wright took her undergrad as well as medical school training at the University of Michigan, graduating with honor Alpha Omega Alpha in 1989. After that, she did her residency at Beth Israel at Northwestern University, returning to Boston after her internal medicine, where she did her pulmonary and critical care fellowship at Beth Israel at the Brigham and Women at Harvard, where she stayed there for additional 3 years of research training, completing that in 1996. She was recruited to the faculty at Harvard, where she stayed until 2010, where she will get promoted to associate professor of medicine. A couple of years later, she was recruited to Mount Sinai, where she remains currently. Those of us here in Madison have had the pleasure of working with Dr. Raz as the co-chair of our executive committee for a major NIH trial, which is the multidisciplinary study to study severe asthma. It's called the Precision Intervention in Severe and Exacerbation Prone Asthma, or PRECISE. And Dr. Wright, she is the co-chair of our executive committee, helping to lead this major NIH study. Dr. Wright is the PI of a number of grants and awards from the NIH, as well as Director of Research Center funded by NHLBI, where she has done research to examine early life predictor of diseases, including asthma. She has had more than 250 publications to date, with particular focus on both the social, like stress, and physical, like air pollution, factors explaining health disparities, particularly in early childhood. Today, Dr. Wright will speak to us on uncovering the links between toxic stress and asthma, implication for disparity, prevention, and treatment. Please help me welcome Dr. Wright to the podium.
Thank you very much. I feel after that introduction, I really want to just say, go blue. Both of my kids went to Michigan too, so we're 4 for 4. It's crazy on football Saturdays. And you have one coming up on Saturday, huh? So it's an absolute pleasure to be invited here. This is my first trip, unbelievably, to Madison, even though I have worked for a number of years with many of you. It's good to see folks in the room, and welcome to those who are still doing this hybrid, but it's just tremendous to be up in front of an audience again. This is my first grand rounds, actually, that way. I've done them virtual for quite a number of years now. So I'm going to talk to you about some of the research that I actually started as a pulmonary fellow in Boston. And I've been really keeping at it since then, like a dog with a bone. So I'll talk to you about some of the work we've been doing in population-based studies to understand independent effects of the social determinants like psychological stress, but also why we need to understand it in the context of the broader environment. And that's kind of where the field has been moving. I have no relevant disclosures to make. The objectives you saw, I hope, that were sent around, we're going to understand that asthma is a disease that causes stress. I think many of clinicians who take care of folks understand that and see that. But also psychological stress is causal. And I think the data is clear. I think when I first started, we thought it was going to be that way. But I'm actually very, 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 very convinced this is something we need to pay attention to. And I'm going to describe some of the biological connections that link psychological stress with asthma onset in particular. and utilize a life course framework. Why am I an internist sitting in a department of pulmonary or pediatrics now? Because I'm interested in understanding where it all begins. And if you know where someone comes from, you have a better understanding of what's going on now. And I think that's a paradigm shift that we're all making in medicine and in this kind of research. So how do we think about it and use a life course framework to do some of this work? Now, for those of you, many of you in the audience know asthma better than I do probably, but it's a disease that has its origins very early in life. Most manifests before the age of, 5 to 8, 50% by age 3, 80% by the age of 8. It's a multifactorial disorder. It's been increasing. We've seen epidemic proportions over the last decades, and that can't be all genetics. So it has to be environment. You know, my original training at the University of Michigan was in genetics. And that's where I thought my career was going to go. But when I started seeing patients, I thought there's something else I need to know here. And that took me back to the public health degree that you heard about. And I'm going to talk specifically about insights in this regard from longitudinal pregnancy cohorts, so epidemiologic studies, and how do you start to think about and measure social determinants? How do you think about it? Is how it might get in the body just as you breathe in an air pollutant or tobacco smoke. Psychological stress gets in there the same way, disrupts similar physiologic systems. So it's not a big stretch. It's a reasonable paradigm to understand stress health links. And then why do we have to look at it along with the other factors? Chemical, physical exposures, biological exposures. A lot of interest now in RSV as we're seeing, that really rise after the COVID pandemic. And then not just risk factors, but the fact that we got to be thinking at the same time about risk and resiliency factors, the balance of those things, because that's how we're really going to translate it to better health outcomes in our patients. So I'm a adult trained pulmonary physician. My clinical care was on the right-hand side of this slide, but how far back, I said to myself, do I need to go where I can really put people back on a healthy, optimal trajectory and that if you got to go back to the root of it, and the root of it really starts where I circle here, prenatal and early childhood, before the onset, before the clinical manifestation. We want to understand what signals can we find that might help us to actually prevent the disease, not only characterize the endotypes and those sorts of things, so we know how to treat it better. Even starts preconception, but that's been a little bit harder to realize. So many of you have probably seen this slide here, because Dr. Lemansky gave this to me many, many years ago. I went up to him when he was giving a talk, and I said, I really want that slide. Can you send it to me? But I said, I'll always have you on the slide. But I could talk about stress in any point in the natural history of the disease. And it's interesting, and there's lots of things to say about it. But again, I'm going to focus really at the left-hand side of this slide and start at the root causes of it. And there are many factors that start to program asthma risk, starting in utero and early childhood, as you see at the base of this tree. And the piece that I thought wasn't being done well, and Dr. Bussey was a major part of bringing that to the point where NHLBI wanted to fund these kinds of studies, right? I remember that workshop way back when. That just set me, was the most thrilling thing I think I had read when I was just starting out. And so and I thought, well, we need to build the evidence base. Where many people were looking at these other factors, but how do we think about measuring stress, and how does psychological stress get under the skin to influence disorders like respiratory health? When I started looking at the literature, it just was so obvious to me that it was the same as when I was reading literature on how air pollution might affect the programming of asthma, how tobacco smoke, how when you breathe in an aeroallergen and it starts to program and change how your immune system and it responds, et cetera. Same systems really are getting pushed and pulled on by psychological stress. And this just gives a cartoon. We all experience challenges in our environment, stressors. We balance that against our coping strategies and supports that we bring to bear to help us rise to the occasion and take on those challenges. And when those get overwhelmed, they overwhelm your ability to cope. It changes your physiology. And the two key systems are the hypothalamic pituitary adrenal axis, so cortisol production is the main outcome, but also the sympathetic adrenal medullary system. And those two talk very much back and forth with the immune system. And asthma is a disease of dysregulated immunity. Makes sense upstream. If these things are also dysregulated, that's going to play a role in the immune function that we're seeing drive asthma. But the other one that's coming more and more clear to me is oxidative stress. And the antioxidant balance, we're seeing more coming out in the asthma literature that says that's a major pathway as well. And when you think about psychological stress, that's also a key player. And I think it's one of the big areas where we need to know more to really understand certain risks, people that are particularly at risk. I think it's the oxidative stress, but it's a balance between all of them. So how do you start understanding that as it might start in utero to program the infant? Well, mom carries stress in from her entire life course. Her systems have already been programmed based on where she's come from, the HPA, autonomic nervous system, her immune function. She carries that into pregnancy and continues to have to manage and face those challenges that come at her. And that's going to get to the baby. It programs the placenta, it comes through the placenta in particular. So inflammation, oxidative stress in the placenta. Oxidative balance in the placenta is so important to normal, optimal fetal development, including the lung. But you could talk about this with almost any disease system. Now, I have the cartoon of the smoking in the bottom with the ultrasound, because most people can wrap their head around if mom's smoking, taking in those toxins, we know it gets to the baby, affects the lungs. Stress is no different to me. I think about it the same way. And I'm not only going to just look at stress, but again, we're going to think about how chemical exposures also interact with stress. And I'm going to focus on air pollution today. We could talk about many of those chemicals. So this was my conception when I started the first, designing the first prospective longitudinal cohort. Let's start in pregnancy. Let's measure all the things we already know are important as rigorously as possible: air pollution, nutrition, diet, go into the home and measure dust and quantify the allergen exposures, tobacco smoke exposure, and then psychological stress. And I put it at the top of this figure now, because over my career as I've done this work, stress, and when you're thinking about health disparities, it rises right to the top. And it's one we have to be thinking about, or we're missing a big part of what's going on. And this was my motivation. This was a this is a study that came out in 1995. And it's a map of the Boston metropolitan area. And it's the different communities and looking at asthma morbidity. As you go from the lighter shade to the darker shade, there's increasing morbidity, no matter what marker you're looking at. And when you look at those communities, they're also the minoritized populations, lower income populations, the people that we're seeing overly burdened by asthma. They were the people that were coming into the hospital. They were people that were getting on the ventilator for asthma and didn't seem to be responding to the treatments that we thought should work. That's what stuck in my head. And why? What's different about these people? So that is somehow click something different. See if I can get back at it. So this was the study. We recruited out of those neighborhoods. We recruited out of community health centers. We didn't go to the Brigham. We didn't go to the Beth Israel Hospital. We went to the community health centers because we wanted not only diversity in the population that we were looking at on the individual level, I wanted to understand maybe there's something about those communities that's different, that's driving it as well. So look at the community level as well. And that was the Asthma Coalition for Community Environment and Social Stress, the ACCESS project, which was one of our first studies. Started recruiting in 2002. And interestingly, my father-in-law drew this cartoon. He was a cartoonist for the Detroit Free Press, gone now, but I remember I asked him, hey, I could do this free, right? Started getting my logos. So one of the first questions we asked of that study, we recruited in pregnancy, we followed moms and babies longitudinally. Are children born to mothers who experience greater stress during pregnancy more likely to develop asthma? And this was one of the first papers that came out in the American Journal of Respiratory Critical Care Medicine, where I became convinced My postdoc came to me and she was looking at a survey measure of negative life events, the crisis measure. It was about 84 items. You could boil it down to stress in 11 life domains, and you could see some of them there: financial strain, trouble with legal issues, neighborhood and personal safety, etc. And we just looked at a negative life event score across the number of life domains that you're experiencing stress. Because, you know, like the little guy on the hill, when I know where I have to step back and do something to get myself back into balance is when too many are piling up, right? Too many things are coming at you at once from all these different, you know, life domains. And that's where you start to experience things are getting out of control, right? You start feeling it physically as well as mentally. Anyway, and that's where you're going to see those systems get pushed and pulled in a different way. And if that's sustained over the long term, you think about people living in some of these lower income, highly impacted environments, it's just constant, and it's too many, and it's too high impact stress. So that's where those systems get reprogrammed and you're going to start to see disease. Anyway, the graph is showing you just from on the bottom of the graphs is just the number, the counts of life domains that you're experiencing stress in. Prenatal on the left-hand side, postnatal in the first 18 months of life, so that early infancy period as well. And we looked at them independently, and you can see there's independent effects of both the pre and postnatal, and it was such a dose-response relationship, controlling for all those other things: air pollutants, allergens we'd measured in the home, tobacco smoke, sociodemographics, etc. I said to the postdoc, there's something wrong there, you gotta go back, take a look. Can't be that amazing. But it always shows that dose response relationship, no matter what tool we use. The other way we looked at it, this is a paper that came out in the in JACI a couple of years later, because as I said, we looked at these in Access, we looked at not only individual level heterogeneity, but this neighborhood level. So we said, where you live matters, and maybe that's driving some of it. So it's not only the individual stressors that we measured with that other tool, but we also looked at community level stressors. And one thing that through focus group work in our populations that came from the people living in the communities, they said to us, what about the crime, the violence? I worry about my kids going to school. There was a lot of gang violence at that time going on. There were random shootings, people going to Pop Warner football practice, and you know, things that I didn't worry about with my kids in the suburbs living in the same area. So we said, okay, how do we measure exposure to violence in these communities? And so there was a validated measure that folks were using at the School of Public Health, and we looked at it in our population. But we also measured another neighborhood factor that people we know is driving some of the respiratory effects, and that was air pollution, traffic-related air pollution in the urban environment. And what this shows is when you put those two measures in the same model, they're in there together, they have independent effects on risk for repeated wheeze, asthmas, we followed them out, lower levels lung function when we followed them out, and we're able to measure that as well. Here we're looking at repeated wheeze by the age of 3 years. And there's a dose response relationship, independent effect of the community violence, the community air pollution from the traffic, and they were the magnitude. The thing I like about it is it shows you the magnitude of that effect is similar. So if we're so concerned about air pollution, why aren't we thinking of this other factor? So it's independent of the air pollution effect. This is another way that we've looked at early life exposure to stress and asthma. And we did it, folks came to me from Denmark and said, hey, we have this great registry data. Everybody who gives birth, we can follow them, we can link to their medical records. And we have data on stress in that we can characterize adverse childhood experiences. That's another way people characterize stress in this early part of life. It's been shown to be linked to lots of chronic diseases. And in this case, it was either loss of a close caregiver, death, or psychological problems in a primary caregiver. And then we looked at it in relationship to asthma phenotype. So early onset asthma, transient, and then people quote unquote outgrew it. By the time you follow these kids to 10 to 11 years, they had late onset and persistent, and then persistent over the whole time period. And you can see boys and girls are similar in their phenotypes. And actually in boys and girls, exposure to the more than one or more ACEs in that early part of life versus none was very predictive and significantly related to whether or not they went on to have these asthma phenotypes. The girls looked like that they were a little bit more impacted on late onset asthma, but all the phenotypes were impacted. So in a huge, huge data set, we were able to characterize and show that too. And that came out more recently in thorax. Now, what about an objective measure? We've been using parent report of asthma, asthma from the electronic health record, a diagnosis from a physician, as we did in the last slide. But now these kids are old enough, 6, 7, 8 years, we can bring them in and get spirometry on them. And so we did bring them in. We used a PC-based spirometer in the laboratory, overreading, very high quality data. And then we looked at relationship to that crisis negative life event measure, the first negative stress measure that I showed you, and whether or not you were low in pregnancy, low in that prenatal environment, that was the referent group, versus exposed at the different time periods, higher levels of stress in the mom, just in pregnancy, only in the early infancy period, or high in both. And you can see there's a sort of a dose response relationship between cumulative exposure in both those periods and lower level lung function at 6 to 7. Why is that important for the pulmonologists in the room? That lung function from age 6, 7 tracks the rest of your life. And it already is saying you're not going to obtain your maximal level that would have been optimal for you to put you on a more healthy trajectory and not be at risk, higher risk for other things as you continue to go on. So it's very important that early period to understand what's getting you there already so that maybe we can intervene. And this was one of the first studies to show that psychological stress, independent of these other factors that we were also able to measure in the cohort, is a predictor of reduced levels of lung function in these kids early on. The other thing that we learned is, this gets a little bit at the life course. Why do we need to have measurement in these repeated measures over time and in these different critical windows of development where we know kids are plastic during pregnancy, but still the lungs are growing all the way through adolescence into early adulthood, right? Here's one that showed the negative life event scale again, predicting repeated wheeze, could be asthma, whoops. I don't know why it's on a timer all of a sudden. And what we saw was boys at the top in the middle panel that's circled, boys had that dose response relationship between increased stress that the mother was reporting during pregnancy and repeated wheeze. Pretty flat when you look at the bottom, right below that for boys for the early life. They're not so vulnerable to the postnatal stress period. They really get the hit early in prenatal. And the girls are flat prenatal, but it's postnatal where they're more vulnerable to the stress effects. And now we're getting a little bit closer to maybe why that is, and using information we're getting from our psychology colleagues and in animal studies. I think it's a different mechanism for the boys and the girls. I think it's really oxidative stress prenatal for the boys, and we'll shoot a little data on that. And I think it's HPA programming more for the girls, and we'll see a little data on that too. Don't know that for sure, but that's sort of what my working hypothesis that is at this point. This is the same pattern that we saw when we followed our access kids for asthma development, asthma diagnoses. You see pretty flat for the girls prenatal, for that cumulative exposure to negative life events in the moms, and they take the hit postnatally. So timing is as important as dose, and that is what you get from a more of a life course longitudinal look at this. We definitely need to be continuing to follow these kids. We need to invest in those kinds of studies and characterize more and more what's going on. So there's still need for those kinds of studies. Now, why do we measure both social and chemical stressors other than, importantly, to show that there's an independent effect of the chemicals that we already knew and already thought we knew are causing asthma, but there's an independent effect. So that's one reason, right? Because a lot of times I'd start these talks early on and my colleagues would just say to me, well, you know, stress tracks with these other badness. And I think it's just correlated and it's just a confounder. And it's an independent risk factor. And that's what we showed by controlling for both in the same model. But what about interactive effects? Because we know they push and pull on the same systems as some of these chemicals. And so air pollution pushes and pulls on the same HPA axis functioning, autonomic nervous system functioning, oxidative stress, as well as ultimately leading to immune dysregulation. They push and pull on the same systems, not completely overlapping in how they do so, what processes are disrupted. So you can see there should be synergistic effects as well. And then you want to know if you're going to design interventions, maybe what's most important or what's the big driver when you're looking at certain populations. So how do we look at air pollution? If you have an address, and this is a very important, a powerful tool in epidemiology. If you have an address on people, you know where they live at these different time periods that you're following them, you can link that address to estimates that are derived for air pollution on a daily level. And I'll show a little bit of that, how that's done by my colleagues who are creating those estimates. And you can get that on a daily level, and you can go back in time because you're using satellite data calibrated to ground monitors from the EPA that's constantly measuring these things to create those estimates. So for my cohort, for example, the satellites went up in 2000 that we're using to create those. I started recruiting in 2002. I can even go back in time, even though I hadn't measured it, you know, physically I hadn't measured it. And I can recreate daily levels of air pollution exposure all the way back to pregnancy, but also even preconception, given the timing. That is a very powerful tool. And you can do it with many, many geographic exposures that are kind of characterized in this bottom right-hand corner, but we're going to focus on air pollution. So we did that. We linked that in Access to where our mothers lived during pregnancy. If they moved, we updated it. And we used the satellite data, like I said, and then you can see in the Northeast in the map on the bottom left-hand corner, those dots are the EPA ground monitors. So there's a lot of them. They're highly concentrated. So you can get good estimates in that area in particular. People have now, you know, developed methods where you can do it even when those are more sparse, but New England was a good place to do it. And this paper has been published too. It came out in the American Journal of Respiratory Critical Care Medicine. And what this is we use data-driven methods. Let the data tell us where the window of vulnerability was. Because we had such highly temporally resolved data, we had daily measures. That's a powerful thing to have in epidemiology. And we looked at it in relationship to asthma development by the time these kids were age 6, 7. And you can see there, when you have a white area above, where you have that shaded area go above the line where it's circled, that's the significant. That says that's the window of vulnerability. That's in mid-pregnancy. And now you can start to hone in and say, well, what are the processes and things going on? You get a little better understanding of what the mechanisms might be. But the interesting thing was, yes, it was independent when we adjusted for all things, including stress, including child sex, all those social demographic factors. But if you actually started looking at interactions, only those kids that were born to mothers that had both high stress and high pollution had that pattern, pretty flat on the left-hand side. No effect of the air pollution if that mother wasn't also experiencing the psychological stress, the increased stress during pregnancy. And it was a three-way interaction. It was really the boys that were most vulnerable. And this was the first clue that we got, that we started thinking maybe it's the oxidative stress, because that's a big driver of the air pollution effects that's affecting boys more during prenatal, and really didn't have much impact on girls' diagnosis of asthma. So then in that same study, we started to do some mechanistic work to try to see if we could get support for what we thought might be going on, for how psychological stress was actually getting in there and driving asthma. One thing we did, and this just reminds us of the different domains that we're interested in looking at. We're going to look at HPA axis function. We're going to look at antioxidant balance as well, or oxidative stress, on some of the things we've done. We've done the others, but we're going to take a little bit of a look at what we did here for HPA axis. What do? We asked our mothers during mid-pregnancy to get us a sample of saliva 5 times a day over 3 typical weekdays. And we sent them home with kits, and we asked them immediately when your eyes open in the morning, because that diurnal curve starts there. Then there's that morning rise in the next 45 minutes, and then it gradually should come down to a nice nadir over the course of the day, that diurnal pattern that we usually think about. And you can see that in the gray shaded area, those were the mother's HPA diurnal rhythms during pregnancy for the kids who went on to develop repeated wheeze. Can do it for asthma, can do it for the lung function we were looking at as well. So it was moms who had hypercortisolism, never really came down to that nadir over the course of the day. Their HPA axis was disrupted. That kid was seeing more cortisol and more implications of that, and their own HPA axis was starting to get programmed in utero. And that seemed to be a big driver of what was going on with asthma. Interestingly, we also collected hair on our moms at delivery. And you get it from, you go under here, nape of the neck, take it at the root. You don't want to pull out the roots, you just cut it right at the scalp. It's not cosmetically a problem. 95% plus women will say, yes, take my hair. And hair grows about 1 centimeter a month. And if they had 9 centimeters of hair at the time they gave birth, we could recreate the whole pregnancy for cortisol production. Now, going back in time again, we didn't collect daily anything to be able to characterize that. We didn't, and it's an integrated measure of that HPA axis. We lose some information, right? We lose the diurnal variation. There's interesting data to be had there. But I show you this slide because if you look at it and you divide by Caucasian, Hispanic, and African Americans in our population, the cortisol levels are quite different across the trimesters in these women. Our African American women, which is in the darker shaded boxes, they came in with the highest level and maintained the highest level of cortisol production over that course, in line with what we were seeing with the mid-pregnancy salivary diurnal rhythms. That was more African Americans too who had that elevated level as well. So it's the physiology that's there that is really driving, I think, a lot of the disparities, and stress has a big part to play there. And you can see down here in the lower right-hand corner, that's a graph that shows, well, what might be driving that. One of our measures we had was a report of history of interpersonal violence exposure over the course of their lives. Child abuse, dating violence during adolescence and adulthood prior to the pregnancy and during the pregnancy. And it was any abuse, emotional, physical, or sexual abuse, and we divided that out. And you can see the African Americans on the right in our population, and surveillance data shows this. And a lot of it's not just race, it's poverty. And it's, living in chronic poverty and being more likely to be exposed to these things. These are universal exposures for sure, but differential exposure to these traumatic kinds of stressors, I think, is driving a lot of what we're seeing. Another biomarker we looked at for oxidative stress was telomere length. And this is just a little cartoon. Telomeres are the protective factors at the end of your chromatin that protects the genes. And oxidative stress shortens those telomeres over time. Many environmental factors do it. Oxidative stress, it's a dosimeter of oxidative stress or chronic oxidative stress, or that's at least how a central way people conceptualize it. What your set point is at the time you're born matters. Kind of like lung function. If you have lower levels of lung function than what you were optimally supposed to have at age 6, 7, you're never going to achieve your optimal. Same thing here. You start with shortened telomeres, you're going to lose those faster over time as you continue to have those, the balance between risk and resiliency factors over life. So that set point at birth is very important. We wanted to know, is stress causing those shortened telomeres at birth? Not a lot of people had been looking at that. And there are several factors that do it. These are data that are published. I won't take a long time to look at this, but again, our boys were most vulnerable here to psychological stress measures in relationship to shortened telomeres at birth, but also the higher levels of cortisol that we know was happening in those populations. That was a big driver of shortened cortisol or shortened telomere length at birth. And specifically in boys, didn't really seem to affect the girls so much. And then you look at another, this is more of a cumulative exposure to oxidative stress than even the telomere length is. If you isolate the mitochondrial DNA, and mitochondria are, the factory that helps us manage oxidative stress, right? So if you get dysfunction in mitochondria, and we're seeing that play out in lots of different health issues, and it's becoming more interesting in the context of asthma risk. What we did was we sequenced the genome for the mitochondrial genes and looked at the mutational load. And this is another index of cumulative oxidative stress. And here we did it in the placenta, which is that important programming organ between that mother and the fetus. And we think a lot of the signaling is coming through there that's programming a lot of what's going on. And these are data that were published in Biological Psychiatry. And we looked at that cumulative lifetime stress measure of a traumatic and non-traumatic stress, a little bit different measure than what I've been talking about, but it's a validated standardized tool to look at lifetime cumulative trauma and non-traumatic stress in these women. And you can see a dose response relationship between increasing exposure to that stress measure and greater mutational load in the placenta. But if you look at it by race on the right-hand side, it's really, again, in our African-American women. Even though our Hispanic women are reporting a significant exposure to these things, somehow they're not taking that hit. That's in green. The African-Americans are in the red. And whites they're seeing a relationship too, as well. So there's a lot of data that says, we know lower income, minoritized populations are experiencing more of these types of stressors, chronic cumulative stress, trauma, as well as non-traumatic stressors, more on average than their higher income non-ethnic minority counterparts. And there's a lot of data to think maybe oxidative stress is driving that. So people have looked at markers of oxidative stress in different biological matrices. And one of the more common to look at were F2 isoprostanes in the urine samples. And we had urine samples on these women. And this is from a different cohort. This is from a Medicaid population in Tennessee, the CANDLE study. And Dr. Keisha Carroll, who's our division chief of general pediatrics, led this project. And we looked at asthma risk in the kids followed to age 4 to 5, 6 in these, and related it to their level of oxidative stress, a biomarker of oxidative stress measured in pregnancy in these mothers. And you would guess that, it's going to be the African-American women that this relationship is going to be seen in, right? They're going to have higher F2 isoprostanes, but that wasn't what was shown. It was only seen in the white women that it was related to an increased risk of asthma. And so people were scratching their heads about that. But if you go into the literature, lots of people are finding in diabetes studies, other studies, that if you also have obesity, that actually lower levels of F2 isoprostane, the 8-iso-PGF2 alpha in particular, which is the gold standard that had been being used, lower levels were more predictive of adverse outcomes, and particularly in African-American obese women. And so we thought, well, maybe what we need to do is look at it in relationship to some of these effect modifiers, but also maybe we need to look at other markers, not just one, but a higher dimensional biomarker. Because a lot of times it's not just one factor. There's a whole host of factors and metabolites that happen, and you adapt to your environment. Maybe we need to have a bigger picture of oxidative stress in these people to really tease this out. So we measure, the folks in our chemistry lab at the Exposome Institute are just amazing at developing newer assays that can be done on smaller and smaller volumes of sample, which is great. And they can do now a panel of oxidative or these F2 isoprostanes and their metabolites on just 100 microliters of sample. And so we took the samples back and we had those rerun. And we not only did the F2 isoprostane, that is the gold standard, but we also took a look at these other metabolites to get a bigger picture of it. And it just shows you the distribution in our African-American, Hispanic, and white population. Really, all of these are higher in our African-American, Hispanic population. But only when we looked at, again, lifetime traumatic stress on the LSCR and risk for childhood asthma, there was a dose-response relationship, but it was only significant in our African-American group in the middle. Really didn't see much at all. We don't show the whites on this graph, but we didn't see much going on there at all. And then we looked at the distribution of the biomarkers, the different oxidative stress biomarkers. And it was really only in the high BMI group on the left-hand panels, two of the different markers, where we saw an increase in relationship to lifetime traumatic and non-traumatic stress. Didn't see much going on in the low BMI group. This is a complicated slide, but it just shows you that there's effect modification by race and BMI, and it was mostly in our African-American obese women where we saw disruption. And then we used a method where we could take a look and create an index of exposure to that mix of oxidative stress biomarkers. And it's called weighted quantile sum regression. We use it all the time in environmental health to look at mixtures of chemicals like metal, multiple metals exposure. And what this tool can give us is it can tell us what are the main factors that are driving the risk within the different subgroups. So what are the major markers that seem to be driving that risk? And it gives you some insight into maybe there's different physiology across these different risk sets. And that's very true. It's shown here. This is a little complicated slide, but basically what this mixture index showed us was what it was really in our Black mothers in the elevated BMI group, those who were overweight and obese versus normal weight, that showed the strongest association. It was about a 6-fold higher association with the increased oxidative stress. And what it highlights to us is we need to start looking at higher dimensional biomarkers to really understand the physiology. It's just like that in immune function, right? If we look at just 4 cytokines, we don't get the full picture because we know there's a cascade of effect of all these things. And it matters to understand how that physiology may differ across these key factors. And we have the tools now, like weighted quantile sum regression, like Bayesian statistics. We have to have our computational scientists as we get more and more of this data to be able to help us sort out what's going on. But those tools are there. We're in an exciting time to be able to do this on a much more complex level, which we all know we need to do. Let's flip it around to say, well, what are the resiliency factors that we might be thinking about, particularly when we're talking about stress driving some of the effects on asthma? Are there resiliency factors that we can hang our hat on that might point to certain interesting interventions and prevention strategies? So can we prevent stress-induced asthma and wheeze? We said, can we look at our data and say anything about factors that might be mitigating the effects of stress? And this was a paper by a psychologist from Stony Brook who was doing her PhD there in psychology. And she reached out to me and asked if she could use the data to have a look at it in this way. She looked at early ACEs, adverse childhood experiences in our kids, followed from birth to 6 months. And it was based on life stressors that moms were reporting. It was based on psychological functioning in the moms, depression, PTSD symptomatology. It was based on financial strain, those 3, and she created a latent construct for that as ACEs and putting people on a continuum. And it predicted, as we would have guessed, significant relationship between increased ACEs and wheeze frequency as we followed these kids out from 6 months to 30 months of age. So increased wheeze frequency in those who had higher levels of the ACEs. We also had taken cortisol in the kids this time. So we had asked moms to collect salivary cortisol at home in the kids, ambulatory, 2 typical weekdays. We didn't do 3 in this case. And then we recreated the rhythms in the kids. And you could see that there was a significant relationship between higher ACEs and disrupted HPA axis already in these kids. They had that atypical higher in the dotted line. higher levels of cortisol produced over the course of the day. And that typically has been related to adverse health outcomes. It was related to increased frequency of wheeze. And you can see it was really the girls that were affected here. Boys, whether they had this more typical steep cortisol slope or a blunted cortisol slope, didn't have any impact on the wheezing frequency as we followed these kids. But in the girls, if they had the blunted cortisol slope, they were having more frequent wheeze. This is a clue that it really is HPA axis programming that's happening in the girls more than the oxidative stress, and something that we need to definitely pursue. Well, if you balance risk and resiliency factors, what could buffer stress? What could keep that HPA axis functioning more typically and keep kids from developing adverse outcomes like wheeze? So we said, what buffers do we have? What do we have in the data that we could look at? One thing we did when we brought kids, those same kids and moms, we bring them in, we've characterized their stress exposure prenatally in that early part of life. We bring them in the laboratory, we hook them up to cardiorespiratory monitoring, we stress challenge them, and we look at their reactivity and their recovery in the face of a stress challenge. We also videotape those, and painstakingly, the psychology students that are working with us use validated tools to characterize supportive caregiving behaviors in the context of that stress, that interaction that we're looking at. It's an objective measure of caregiving behaviors in the mom to the baby. And that social supports and social networks in your caregiving environment when you're an infant is arguably the most important social support network that you have. That's been shown to be protective against significant trauma and stress rocking that HPA axis. And so we said, well, let's use that objective measure and put it into our model and see if it buffers the effect of ACEs. This actually came to me from Megan Gunner, who many of you may know, who does a lot of this work. And she gave me this slide to show that that caregiving environment is really one of the most important environments for development of kids' emotion regulation and all of those things that track with them. These data are complicated. They are published in Child Development. And what it showed was that sensitive caregiving did buffer, but it only buffered in the boys. The girls, somehow their HPA axis had already been so rooted that the measure of caregiving sensitivity didn't have any impact on them, and they still had increased wheeze and increased disruption related to that increased disruption of the HPA axis. That was a paper that I'm just fascinated by that, and I really want to go after that in a more significant way. We're not going to talk about it, but prenatal diet is another lever that we're going after that can buffer it. If you think oxidative stress, increased inflammation, disruption of polyunsaturated fatty acids, those sorts of things, which we know program the brain, but also the lung, that's another lever that we could start to look at. And we have started to look at that. And I'm just gonna, in the interest of time, because I want to leave time for questions. So this is sort of how we see it. People, you're developing, your lungs are developing, all these interrelated systems are developing like that railroad track, and you're the one trying to keep those trains from running into each other, keep people on track of where they're supposed to be, and what those levers are that can balance, what are the resiliency factors that can balance the risk from these things that we're looking at, and keep that train from running off the edge. Keep kids from having low levels of lung function at age 6 to 7 that already put them behind the 8 ball for increased risk of ongoing problems over there. I think it's nutrition. I think it's social supports when you're talking about buffering stress, but we're also looking at the caregiving for buffering effects of multiple metal exposures in utero that these kids are seeing. And that definitely tracks with your SES, where you live, and minoritized status. Air pollution exposures are programming the HPA axis and all those things as well. Nutrition and social supports may be 2 ways that we haven't been thinking about so much that could buffer that. So now my picture goes much bigger than that circle I showed you at the beginning when I started thinking about this. We know many factors are driving programming of asthma, right? And stress and how you measure it is just down there. I've shown you a little bit of how we've done it, but it's complicated. It's a mixture. It's a mixture just like air pollution is a mixture. It's a mixture just like toxic exposure to tobacco smoke and what comes with that, what chemicals are in there is a mixture, allergen exposures. We are developing novel tools on the left that allow us to embrace that complexity, measure lots of these things at the same time and cost effectively, a little bit more cost effectively so that we can look at them more you don't have to, we're not going to have to do it all and be perfect to get the answers, go a long ways towards those answers. So this is the picture we use now. And really we're going to have to do omics level integrated network science to really tease this out. Because as you do that and embrace that complexity more, you get a little closer to the answer. You uncover something that you weren't even seeing in data before you started to take that more complex look. And that's what we're trying to do at the Institute for Exposomics. And we put it into the life course framework that we've been talking about. And this is something I want to draw your attention to, because we don't have time today to get into that full complexity. But one thing we did through the CTSA network and the Life Course Enterprise Committee there was, how do we disseminate the tools and the expertise that's already out there to help us really go in this direction? Let's make those tools more readily available and be able to be disseminated. It's something that you can go to this link and look at, and in your own time, decide you're going to look at these different modules that we've developed, educational modules. We use them in our coursework to teach students life course research. And it goes all the way from how do we integrate data sets across the lifespan? How do we take advantage of extant data sets right now to start to ask and answer these types of life course questions? ECHO, the environmental influences on child health outcomes, the national program that really brought together 80 some cohorts across the country. And many of us here have been struggling with how to harmonize the data across those different studies that had been designed separately, to all the way to how do we expand traditional epidemiologic methods to include systems and network modeling. And there's a link there, you can go to it. There's a series of experts that had given these talks and provided those to that program. There are TED style talks that kind of get you just thinking, and then they go a little deeper when you get out to that outer area. These are excellent, excellent talks. That was just like so amazingly fun to be a part of that. So I really wanted to make sure that you're aware of that resource, because it's easier to build strong children than repair broken men. I felt like I was, as a pulmonary critical care physician, 4th, 5th, 6th decade, I couldn't move the needle all that much. And how far back do we need to understand where these folks came from to really do better? Identify risk groups, identify endotypes, identify the best therapies that are going to have. We've made huge progress, but we all know there's a lot of work there for things like asthma, and Precise is one of the networks that's trying to do that. And, takes a village. So these are just a few of the folks that touched on some of the things that I talked to you today. I have had a whole host, and I've been, blessed with all the pre and postdoctoral students who want to come do this work, and fortunate enough to get funding to keep it going. So thank you very much. And I wanted to leave time for questions.
Thank you very, very much. That was an outstanding talk, Dr. Wright, and we are grateful for that. Question from the audience, Dr. Bussey.
Yeah, Roz, thank you very much. I mean, you've really made tremendous progress in identifying the importance of all these stress-related factors and also moving into the preventive aspects. Question I have is, you've indicated some differences between boys and girls. What are the mechanisms by which the boys and the girls are affected differently and at different times in their life? Do you have insight into that?
Yeah, I mean, you go all the way back in utero, and if you start reading about the placenta is such a sexually dimorphic organism. If you look at epigenetics in the placenta, that's another thing we've done. I didn't present on that today, but the epigenetic marks between the girl and boy placenta are so different. So there's a lot to be learned there. What gene pathways are being turned on and off in different ways for girls versus boys? That's one thing. And a lot of it happens at that molecular level. one of the things we've started looking at with metabolomics is sort of in mom's serum, in mom's urine during pregnancy, just doing an untargeted scan of metabolomics. We can actually see boys and girls are quite different when you're looking at hormone profiles. If you pull out the different hormone, androgens, estrogens, progestins, and we need to understand that. I think hormonal disruption beyond cortisol, because it's HPA, but it's also HPG axis that gets disrupted by stress just as much. But not only stress, we've published on preconception air pollution exposure, really programs that hormonal milieu that kids are developing in, and it's going to be very different for boys and girls. I think it's been worked out a little bit better in the brain, that girls and boys develop their timing of development of the different parts of the brain are very different across girls and boys. We need to understand that a little bit better for the lung than we do. But a lot of it's to the milieu is different because you're carrying a male versus a female fetus. All of a sudden that shifts what's happening in that maternal milieu that we are looking at. But we need to characterize it more. And that's where you get into the multi-omics and the systems biology, right? Because a lot of times if we just look at a little piece of it, you miss something. Kind of the story with the oxidative stress that we've learned, I think. And that's what I was trying to exemplify by sharing that a little bit today.
There's a question from Dr. Graham. I'm going to ask you, Roz, if you could repeat the question for the recording after he asked the question. So
Yes.
Thank you.
Wondering what's been done in that really area for future studies.
So the question was related to the nutrition work on perhaps being able to mitigate effects of toxins like stress, and how far are we along with that kind of research. I was surprised. We did the food frequency questionnaire, and you know, I had a postdoc working on that, and she had spent a lot of time characterizing the different micronutrient levels, et cetera. And I said, well, maybe we should just publish the, you know, has anybody done that in a lower income ethnically mixed population? Let's see what's out there. There were zero studies on nutritional characterization in pregnant women. Hadn't been done in a long time, hadn't been done since we fortified foods with folate and folic acid and other things. There were lots of reasons why we needed to do it. And that's one of the reasons ECHO's gone after this too, in an even broader population, because there are clearly socio-demographic drivers of food inadequacies and actually elevated intakes that have health implications. So we've done it by, you know, creating oxidant or antioxidant indices and seeing if that protects. We have published on that. It does protect against air pollution effects on wheeze. It does protect against stress on wheeze. But it's only one part of it. And what we've gone, and what we're trying to think about, and ECHO provides a very unique opportunity to do that, because they have food data, micronutrient intake data on thousands of women, you know, over 10,000 women. So think of what you can do with that now. Is to think about it more as a mixture, not just use these standardized indices that have been created in the nutrition literature. It only takes you so far, but to use some of our machine learning and artificial intelligence. We've got hundreds of micronutrients. Let's let the data tell us where some of the factors are that might be helping. And it might be different in your population, your part of what's going on in ECHO versus, you know, my part of the world, or that's the other interesting thing about ECHO. Now we have across the US in different areas, and the social demographics get even more broad. We're going to be able to get a little bit closer to those risk groups and how nutrition and what it seems to be driving it in New York City versus, you know, here in Madison, what's your access to healthy foods, those kinds of things. But even there, I think we got to do it on this big scale, or we're just, you know, we'll find things, but not that much closer to the answer.
So there is an online question from Dr. Teodrescu about sleep, which is an important factor that can be manipulated to mitigate stress. Any data on that?
Not a lot of data on prenatal programming of sleep. In particular, when we went to the literature, And interestingly, we had, and Echo's doing that again. They're getting actigraphy on kids, so you can measure sleep that way when they're getting survey tools on sleep behaviors in kids. That's important. We have an RO1 where we do that, but we also go in and do home sleep studies, PSGs. We can go even deeper. So we've done that, and unfortunately, we started that right before COVID. So put us on hiatus for a little bit. But we've been able to go into and get PSG, home PSG, full montage. Because I trained not only in pulmonary critical care, but I was training in sleep. Because, you know, I was gaming, what am I going to be? I want to be marketable when I get out of here. And I'm always like a kid in a candy store. Yeah, I'll learn something new. Anyway, I've taken it to the lab, but we do home full montage sleep studies, and we have it on about 175 of these kids. We're going to get it on hopefully up to 500 of these kids. Together with the actigraphy data, we can look at circadian rhythm disruption. There's so many things we can look at. So that is a great question. I think it's an unmet, need for research, and we need to do more of it, particularly in that prenatal early life environment. Most people start, when they've already got sleep problems.
Dan?
Thanks, Ross. Really nice talk. So as we, ultimately hope to move the needle on stress, it's obviously huge societal changes that need to be made.
Absolutely right. I mean, we have, ultimately it's policy and upstream. I mean, I was at an NIH workshop on asthma. Oh, I'm sorry, repeat the question. What was the question? In order to really address what's driving a lot of the stressors that we were talking about, you really have to go upstream. Policy, reduce poverty, give people more opportunity. Neighborhoods should not be living with such crime and violence. And where does that come from? It's not the individuals, it's the lack of resources that brings those things to them. So yes, it has to be policy level changes that have the big impact. What can we do at the individual level, though, in the meantime? Because that takes time, right? It takes time even to have the air pollution data say this should be the new set point for air pollution, because we're already seeing health effects. Just think how long that takes. And I was actually at an NIH workshop on asthma once where they said, you get one thing that's going to cure asthma. Everybody stand up and say it. People say vitamin D, this. I said, fix poverty. It would hit us a million ways, but that's going to have the biggest bang for the buck. So how do we build resilience? A little bit how we pivoted to nutrition, right? Nutrition is not something that we'd learn as doctors. It's not something that's done routinely in our clinics. If we can show that there are nutritional, you can tweak nutrition, give people access to healthy foods, and what might that be, and identify for that individual what might help. That's one way we can do now. For building social supports is another. That's why we thought about the caregiving environment. There are validated intervention methods to teach supportive caregiving in that early period that psychologists have worked out to really keep that HPA axis and other regulatory systems functioning well in kids. So that seems to be another evidence-based approach to take. And I find as a clinician, when I just validate for somebody that this is important, and I'm not saying this is in your head. You have to be very careful how you talk to people, because some people get very upset. You're telling me it's my fault for being stressed, and how can I go to my environment and not be stressed when I have bars on my windows, gunshots are outside my window. I worry every day about the mold growing because you guys are telling us that's not good for my kids' asthma, but there's nothing I can do about it. You know what I mean? But you validate for them, and let's take it apart and let's do what we can together. And people probably have other ideas. I mean, these are the things we're going to go after. But it is very important for clinicians to understand this is something we should be talking to people about, just like tobacco smoke, just like these other factors that we're talking to them about. Stress is a huge player, and we do need to figure it out.
And on that note, we thank you so very much for a wonderful talk. Appreciate it. Thank you for attending.